With the genome aligned track loaded, we can load our data and ask our questions of interest. Here we have the human genome H238 as the reference genome, and the mouse MM10 as the secondary genome. We also have gene and repeat annotations mapped to both genomes. Now we can start loading our data either from a local file system or a URL. Let's start by loading our data from a URL. Click Tracks and Remote Tracks. Here we are using RNA-seq of human liver from ENCODE. Next, let's load our data from a local file system. Click Tracks, Load Tracks, and select the track we want to load. Here we are loading an RNA-seq of mouse liver from our desktop that we have downloaded from ENCODE. We will specify the track type as bigwig and the assembly as MM10. Next, click Choose Files to select the file. Click the X in the top right corner to close the window. To better compare the data, we can reorder the tracks. For example, we can group the tracks by species and separated by the genome align track. To reorder the tracks, click the Reorder tool and drag the human track to the top of the genome align track. The browser allows for navigation of the reference genome using either a gene name, a SNP, or coordinates directly. Click the coordinates box at the top to enter the navigation window. Let's navigate to the gene SPP2. We can use the buttons at the top under Tools to zoom in and out. There are different buttons to zoom in or out with different resolutions, or to pan left or right. For example, to zoom out, click the -1 button. It is possible to zoom in to a particular region using the Zoom In tool. Click the Zoom In tool and use the mouse to click and drag over the region of interest. Here we are zooming in to the SPP2 gene's promoter region. Now the browser displays human and mouse liver RNA-seq over the SPP2 gene's promoter region.